Understanding Glycolysis, a comprehensive guide to glucose metabolism. Hello, welcome to the Science Guide ASAP Learning. Today we are going to be speaking about glycolysis. Hope this helps. Please support this new channel by liking this video and subscribing for more videos like this one. Glycolysis is a metabolic pathway that occurs in the cytoplasm of cells and is responsible for breaking down glucose into 2 molecules of pyruvate. The process of glycolysis consists of 10 enzyme-catalyzed reactions, each of which plays a critical role in the overall process. The reactions of glycolysis can be divided into 2 main phases, the energy investment phase and the energy payoff phase. Let's take a closer look at each of these phases. Energy investment phase. The first phase of glycolysis, the energy investment phase, requires the input of 2 ATP molecules. In this phase, glucose is converted into fructose 1,6-bisphosphate, a 6-carbon sugar, through a series of enzymatic reactions. The first step in this process is the phosphorylation of glucose by the enzyme hexokinase, which uses 1 molecule of ATP to add a phosphate group to glucose, forming glucose 6-phosphate. This phosphorylation is important because it helps to trap glucose inside the cell by preventing it from leaving through the membrane. The next step in the energy investment phase is the conversion of glucose 6-phosphate into fructose 6-phosphate by the enzyme glucose 6-phosphate isomerase. This is followed by the phosphorylation of fructose 6-phosphate by the enzyme phosphofructokinase, which uses another molecule of ATP to add a second phosphate group, forming fructose 1,6-bisphosphate. Energy payoff phase. The second phase of glycolysis, the energy payoff phase, produces 4 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate. In the first reaction of this phase, fructose 1,6-bisphosphate is cleaved into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, by the enzyme aldolase. Dihydroxyacetone phosphate is then converted into glyceraldehyde 3-phosphate by the enzyme triose-phosphate isomerase. From this point on, each molecule of glyceraldehyde 3-phosphate undergoes the same set of reactions, resulting in the production of 2 molecules of pyruvate. The first step in this process is the oxidation of glyceraldehyde 3-phosphate by the enzyme glyceraldehyde 3-phosphate dehydrogenase, which uses NAD+ as an electron acceptor to produce NADH in the molecule of 1,3-bisphosphoglycerate. The next step is the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate by the enzyme phosphoglycerate kinase, which transfers a phosphate group from 1,3-bisphosphoglycerate to ADP, producing ATP. This reaction produces 2 molecules of ATP per molecule of glyceraldehyde 3-phosphate. The final step in glycolysis is the conversion of phosphoenol pyruvate into pyruvate by the enzyme pyruvate kinase, which transfers a phosphate group from phosphoenol pyruvate to ADP, producing ATP and pyruvate. This reaction produces 2 molecules of ATP per molecule of phosphoenol pyruvate, for a total of 4 ATP molecules per molecule of glucose. In summary, glycolysis is a metabolic pathway that breaks down glucose into 2 molecules of pyruvate. The process consists of 10 enzyme-catalyzed reactions, which can be divided into the energy investment phase and the energy payoff phase. The energy investment phase requires the input of 2 ATP molecules, while the energy payoff phase produces 4 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate. Glycolysis is an important process in cellular respiration, as it provides the starting material for the production of ATP through the process of oxidative phosphorylation. Hope this helps. Please support this new channel by liking this video and subscribing for more videos like this one.